Here again is a molecular list of ingredients of the body. Each of these compounds can be obtained from our food, but we can also manufacture each of these compounds inside our body. We are chemical factories that can make and break a wide variety of chemical compounds at remarkable speed and low temperature. For example, most of the water in our body comes from outside the body. However, a typical adult will produce at least a cup of water each day as a byproduct of various chemical reactions. When we manufacture nucleic acids, proteins, and ATP, we also make water. Our bodies cannot manufacture all the water it needs to keep us alive, so be sure to take some bottled water with you on your trip to the desert. Much of the fat in our body comes from fat in our food, but we can also make fat by consuming too many sugar calories and alcohol. Fat can hold more than twice the energy per gram as carbohydrate or protein, so our body prefers to store energy long-term as fat. This keeps us lighter on our toes than if we stored the same amount of energy as carbohydrate or protein. We're about 15% by weight protein, all of which was made inside our body. The protein in our diet is broken down to amino acids in the digestive tract, and these amino acids are taken into the body and used to construct our own body proteins. Nucleic acids include DNA and RNA. All are manufactured inside our body with the help of molecular sewing machines called DNA polymerase and RNA polymerase. The carbohydrate content of our body originates both from our diet and from synthesis within our body. If our diet lacks carbohydrate, we can manufacture what we need mostly from amino acids. Any respectable chemical lab must be envious of our body's amazing chemical wizardry. Let's see how we do it. The agents of chemical change in our body are enzymes. Enzymes are like tiny molecular scissors and sewing machines that can rapidly disassemble or construct the many compounds found within us. Enzymes accomplish their chemical feats by providing an alternative pathway for a reaction that requires less energy and so can proceed at remarkable speeds and relatively low body temperature. It's as if you need to get to a destination today, but it would take you a couple of days to get there by car. So you take a plane instead. The plane gets you to your destination more quickly. It acts as an enzyme or catalyst, speeding up your transport. To accelerate chemical reactions, enzymes must bind to their substrates, which is the generic name for the chemical agents on which they act. Enzymes must come into physical contact with their substrates. They do not work from a distance. They do not phone it in. The enzyme shown here is in green. The substrate is shown in purple. The enzyme binds to its substrate, activates it, and then splits it in two. The enzyme then releases the products and is free to do the same reaction again. Enzymes bind their substrates at a special surface called the active site, also known as the catalytic site. The substrate binds to the enzyme at this site in a similar manner to a key binding to a lock. Not every key will fit into any lock, and even if it does, it won't necessarily open the lock. There must be a very specific compatibility between the key and the lock for a successful mating to occur. The same is true of an enzyme and its substrate. To successfully bind, there must be compatibility of shape and charge between an enzyme and its substrate. This substrate has a projection here that fits within a cavity on the enzyme. The substrate has a cavity here that fits into a projection on the enzyme. The substrate has a projection here that fits within a cavity on the enzyme. So we have a compatibility of shape between the enzyme and its substrate. But we also have compatibility of charge. In electricity, opposite charges attract, like charges repel. Note that where we have a positive charge on the substrate, we have a negative charge on the enzyme. Positive charge on the substrate, negative charge on the enzyme. Negative charge on the substrate, positive charge on the enzyme. So the conditions are right for successful binding and reaction between the enzyme and substrate. Once bound, the enzyme activates the substrate, sort of like your chiropractor giving you a bear hug to crack your back. And the substrate is transformed into the product and released. Here you see a digestive enzyme in action. This enzyme binds its substrate at its catalytic site. The enzyme activates the substrate, which splits in two, and the enzyme releases it, and can return to do the same reaction again with another substrate. Enzymes are like scissors. Cutting out a coupon with your scissors does not destroy the scissors. They can be used repeatedly for the same reaction. Eventually, they will wear out and need to be replaced, but not after catalyzing many cuttings. The same is true of enzymes. This story from ancient Persia may help you appreciate enzymes. It begins with a farmer who died and left his estate to his 3 sons. In his will, he indicated that son number 1 should receive half of the estate, son number 2 should get 1/3 of the estate, and son number 3 should get only 1/9 of the estate. The problem, though, is that there were only 17 camels in the estate. Can you see the problem? Half of 17 camels is 8 camels and a lot of camel burgers. Fortunately, along came a wise man. He offered to loan the estate one of his camels. That made 18 camels in the estate. Half of 18 camels is 9 camels for son number 1. 1/3 of 18 is 6 camels for son number 2. 1/9 of 18 is 2 camels for son number 3. So the 17 camels were distributed to the sons without sacrificing any camels. And the wise man got his camel back. That's why he was called a wise man. If he'd gotten 2 camels back, he would have been an entrepreneur. But the camel that he loaned to the estate made the transaction possible. Yet it wasn't changed as a result of the reaction. The wise man's camel acted like an enzyme. Another characteristic of enzymes is that they are not promiscuous. An enzyme will not bind just any old substrate, or catalyze any type of reaction. Enzymes are very particular in what they bind and what they can do to a substrate. They are often named for the substrate they bind or the kind of reaction they catalyze. For example, enzymes that bind and break down starch are called amylases. Amyl is Greek for starch. The suffix ase tells you it's an enzyme. Most enzyme names end in ase. Amylases will break down starch, but they won't break down proteins. To break down proteins, you need a separate group of enzymes called proteases. Proteases, however, will not break down lipids or fats. Instead, lipids require their own group of enzymes called lipases. Lipid is from the Greek word for fat. Lipases will break down lipids, but they won't break down nucleic acids. Instead, we need a separate group of enzymes called nucleases to break down DNA and RNA. To show you how specific enzymes can be, we have 3 major disaccharides, or double sugars, in our diet: sucrose, lactose, and maltose. Even though these 3 compounds are very similar, each requires its own different enzyme to break it down to its monosaccharide building blocks. Sucrose is known as table sugar. It's that white crystalline stuff in your sugar bowl at home. Sucrose requires the enzyme sucrase, manufactured by the small intestine, to break it down into its monosaccharide building blocks. Sucrase will break down sucrose, but it won't break down milk sugar, lactose. Instead, lactose requires the enzyme lactase to break it down to its monosaccharide building blocks. Lactase will digest lactose, but not maltose or malt sugar. Instead, maltose requires its own special enzyme called maltase to break it into its monosaccharide building blocks. Many enzymes are named after what they do to a substrate. For example, hydroxylase enzymes transfer a hydroxyl group to a substrate. This dash here represents a covalent bond, not a charge. Phosphorylase enzymes transfer a phosphate group to a substrate. Again, the dash indicates a covalent bond. This is a negative sign. Methylase enzymes transfer a methyl group to a substrate. A methyl group is CH sub 3. Carboxylase enzymes transfer a carboxylic acid group, COOH, to the substrate that it binds. Many enzymes need help to bind and transform their substrates. One group of helpers are called cofactors and include ionized elements, often referred to as minerals in nutrition textbooks. For example, more than 300 enzymes in the body require magnesium ions to help them bind and catalyze their substrate. This includes DNA polymerase, the enzyme that replicates DNA. Note this enzyme has a negative charge on its catalytic site. If the substrate also has a negative charge, then the two would not bind. But if we fit a divalent cation, like magnesium, into the catalytic site, it will change the electrical surface of the enzyme to make it compatible with the substrate. Many of the minerals in our diet are needed for this particular purpose. A second group of nutrients called vitamins also assist enzymes. These organic compounds are called coenzymes. For example, vitamin C is needed by enzymes that manufacture the most abundant structural protein in our body, collagen. Vitamin C assists in the transfer of hydroxyl groups to amino acids within collagen. Without vitamin C, your enzymes can't make collagen and your body literally disintegrates. The condition is called scurvy. A paperclip needs a particular structure to perform its function of holding pages together. Likewise, an enzyme needs a particular structure to bind its substrate and catalyze its reaction. Change the structure, and you can change the function. Remember what happened to our frog when we ran him through a blender? Enzyme structure is sensitive to environmental conditions, especially temperature and acidity. One reason we need to regulate body temperature and the pH of body fluids is to keep our enzymes intact and functioning. Perhaps you've fried an egg. Raw egg white is a transparent liquid containing globular proteins. But when you drop the egg on a hot skillet, you can see a transformation of the transparent liquid into an opaque solid. The heat is causing the globular proteins to denature or unfold, and all the king's horses and all the king's men can't put it back into the shell again. So you eat it. Elevated body temperature can kill because it can cook our enzymes, causing them to denature. We must also regulate the pH of body fluids for the same reason. If you dropped a raw egg into vinegar, a similar reaction would occur. The egg proteins will denature. The pH of our body fluids is near 7. The pH of vinegar is about 2.5. This high acidity of vinegar attacks the bonds holding the proteins in their three-dimensional structure. The proteins denature. If these proteins were enzymes, their catalytic function would be lost. Another characteristic of enzymes is that they usually work as a team. They are not loners. We call enzymes that work together to produce a product a metabolic pathway. One of the reasons we have so many compartments inside the cell is so we can house together the enzymes that are part of the same metabolic pathway. A given enzyme can make only a small change to a substrate. If we want to make major changes, we need several enzymes. Let's say we wanted to make epinephrine, also called adrenaline. We start out with an amino acid called phenylalanine. As you can see, phenylalanine looks nothing like epinephrine. So we're going to need 5 different enzymes to convert phenylalanine to epinephrine. The first enzyme is called phenylalanine hydroxylase because it adds a hydroxyl group, an OH group, to phenylalanine, which makes another amino acid called tyrosine. The enzyme tyrosine hydroxylase uses tyrosine as a substrate to add another hydroxyl group to make DOPA, which stands for dihydroxyphenylalanine. implying we've now added 2 hydroxyl groups to phenylalanine. DOPA binds to the enzyme D-carboxylase to produce dopamine. The D-carboxylase enzyme removes a carboxyl group from DOPA to make dopamine. Dopamine then binds to the enzyme dopamine hydroxylase to add yet another hydroxyl group to manufacture norepinephrine, also called noradrenaline. In many cells, that's where the pathway stops. But in cells of the adrenal medulla, there's yet another enzyme called norepinephrine methyltransferase that adds a methyl group, CH3, to norepinephrine to make our end product, epinephrine. So we've added 3 hydroxyl groups, 1, 2, 3, and a methyl group, and removed a carboxylic acid group to convert phenylalanine into epinephrine. We produce epinephrine primarily in the adrenal glands above our kidneys. That's where the enzymes for this metabolic pathway are housed. If we're wrestling with the boogeyman or running for our life, we need lots of adrenaline. If you're taking your afternoon siesta, you don't need much adrenaline. Somehow, the body must be able to regulate the production of this hormone based on our needs. We can do that in part through what is called end product feedback inhibition. You're viewing a generic metabolic pathway involving 3 enzymes, 1, 2, and 3. What this pathway suggests is that enzyme 1 binds substrate A at its catalytic site, modifies it, and releases it as B. B is then picked up by enzyme 2, modified, and then released as C. Enzyme 3 then picks up substrate C, modifies it, and releases it as the end product of the metabolic pathway, D. Note that enzyme 1 has 2 binding sites. The catalytic site is for binding the substrate A, but it has a second binding site called a regulatory site or allosteric site that doesn't bind the substrate. Instead, it binds the end product of the metabolic pathway, D. This second site is used to regulate the production of D by the metabolic pathway. It's like an on-off switch. When the site is open, the enzyme is on, converting A into B. But as the end product, D, accumulates in the cell, there is a greater probability it will find its way back to the regulatory site of enzyme 1 and bind to it. When the end product binds to the regulatory site, that changes the shape of the enzyme so that its catalytic site will no longer bind substrate A. And if enzyme 1 can't bind substrate A, it can't make B. And if it doesn't make B, enzyme 2 can't make C. And if there's no C, enzyme 3 has nothing to use to make the end product D. So the end product in the pathway will no longer be manufactured. This is called end product feedback inhibition. As the end product accumulates, there's a greater probability it will find its way to the regulatory site of the regulatory enzyme, which is enzyme 1, bind to it reversibly, shutting down the enzyme so it no longer binds its substrate. This prevents the overproduction of the end product. As the end product gets used up, however, it pops off the regulatory site, and as a result, the catalytic site is restored and can begin the reaction again. It can begin to bind A and convert it into B, which then makes B available for enzyme 2 to make C, which makes C available for enzyme 3 to once again manufacture the end product. Here's an animation of a metabolic pathway in action. We have 4 enzymes in this metabolic pathway. Enzyme 1 will bind substrate 1 to make substrate 2, which binds to enzyme 2, which makes substrate 3, which binds to enzyme 3, which makes substrate 4, and then substrate 4 is converted by enzyme 4 into the end product. It looks like we're making jelly beans. So this reaction is occurring between the substrates and the catalytic sites of these enzymes. One enzyme makes a change in the substrate and then passes it along to the next enzyme in the chain. Now notice the jelly beans are accumulating in the cell, and we don't want jelly beans running out of our ear, so notice what's happening. As they reach a critical quantity, they'll find their way to the second site of the first enzyme, the regulatory site or allosteric site. And when the end product binds here, it changes the shape of the catalytic site of the enzyme so it no longer will bind the substrate. As a result, it will stop the chemical reaction, stopping the production of the end product. Then when the jelly beans get used up in the cell, they'll pop off the regulatory site and start the reaction going again. So here again we have the production of jelly beans in progress. Here's our allosteric site, currently unoccupied in the first enzyme. Now we're starting to accumulate jelly beans in our cell. Making too many jelly beans is a waste of resources, so we don't want to do that. So one way we have of preventing that is end product feedback inhibition. And there goes a jelly bean finding its way back to the regulatory site of the first enzyme, and notice what happens up here. The catalytic site changes, closes off so that it will no longer bind the substrate. Recall that our cells use dietary fuel molecules to produce ATP, which is the form of energy needed by cells to do work. Most of our ATP production occurs in the mitochondria, which houses the enzymes and transport proteins needed to complete the transfer of energy from dietary fuel molecules. These fuel molecules, primarily glucose and fatty acids, are introduced 2 carbons at a time into the mitochondrial metabolic pathway known as the citric acid cycle, or Krebs cycle. Exiting the mitochondria are byproducts, carbon dioxide and water, and the critical end product of the metabolic pathway, ATP. Our need for ATP varies considerably throughout the day. When you're napping, you need very little ATP. When you're running a marathon, you need a lot of ATP. If we measured the ATP levels in your muscle cells, we would find very little difference in concentration whether you were napping or exercising. This implies that we can adjust the rate of production of ATP to match the demands of the moment. How do we do this? There are at least 3 levels of regulation involved in the production of ATP. The first is end product inhibition. Some of the enzymes in the metabolic pathway that produce ATP are sensitive to ATP levels in the cell. If ATP levels rise, there is a greater probability ATP will bind to the regulatory sites of these enzymes and shut down ATP production. As ATP becomes scarce, the ATP pops off the regulatory sites and the pathway is reactivated. This level of regulation is extremely fast, effective in a matter of seconds. The second level of regulation involves production of additional enzymes for the ATP metabolic pathway. This requires genes coding for these enzymes to be switched on. This will take several hours or days to become effective. The third level of regulation involves producing additional mitochondria. Recall that mitochondria can divide independent of the cell. If you're a couch potato, you may have only 100 mitochondria in a particular muscle cell. But if you train to run the Boston Marathon, you may have several hundred mitochondria in the same cell on race day. You can't increase the number of mitochondria in your cells overnight. You have to train for weeks. By doing so, you are in effect manufacturing more power plants to make the fuel you need to support your customary level of activity.